We are so pleased that our next speaker, Dr. Frank Szoka, was able to join us today. Frank's experiences demonstrate the classical path many academic scientists take to commercialize their inventions. He is currently a professor of bioengineering, therapeutic science, and pharmaceutical chemistry at University of California School of Pharmacy in San Francisco. As a serial inventor and entrepreneur, Frank holds 27 U.S. patents and has founded several biotech companies, including Sequus Pharmaceuticals, Gene Medicine, and Zone 1 Pharma. He received a Bachelor of Arts in Liberal Arts at Johns Hopkins University, a master's degree in microbiology from University of Maryland, and a PhD in biochemistry from SUNY Buffalo. Please join me in welcoming Dr. Frank Szoka.
I got it right there.
Is this one? Is that it? Okay. Okay, and then there should be a little, damn, these screens are small. Okay, I am absolutely delighted to be here, and it's nice coming back to Boston. I thank the President Larson of your university for welcoming all of us. I was wondering, is she related to Gary Larson, who has a scientific bent to his cartoons? What I'm going to do is give you my personal experience at being involved in science and translating or trying to translate that into products. And it started a long time ago, and these are the destinations that I'm going to tell you about in terms of my vacation. Lipis Ohm technology in 1981, which became Sequus. 1984, Gene Medicine, and that's where I first met Fred, who was also a founder of Gene Medicine. And finally, most recently, Zone 1 Pharma. So I've gone to Menlo Park, Houston, and now I'm back in San Francisco in terms of companies. And so LTI was conceived in Buffalo. And the person in the upper part of the screen is Dimitri Papahajopoulos. And Dimitri was my mentor as a fellow. And so I got my PhD and I went to Roswell Park and I worked with Dimitri, where I actually did biophysics. And he was much like the Bruce's mentor, that you could talk to him, he'd give you ideas, and the two of us filed some patents at that time, thinking that we could turn them into a company. And this was in the late 70s. And it was based upon basically NIH-funded science, and these are just three of many that we published that all related to lipid vesicles. And I'll tell you what a lipid vesicle is in a little while, but it was this technology that led to a series of patents that we had with us when Dimitri moved from Roswell Park to the University of California at San Francisco. And he did this in 1978, and there he was reunited with an old friend from Greece by the name of Nick Arvanitidis. And Nick is up in the right-hand corner there. And so Nick had been on the West Coast running a public sector consulting company. And Dimitri and Nick got together, and over a backgammon game and strong coffee, they said, geez, biotech is coming about. Do you have anything that you could form a company with? And Nick said, yeah, I have a really bright postdoc. He's gone off to Boston. By that time, I had moved to Tufts Medical School, where I was in the physiology department as a professor. And he said, we have some technology that we've patented. And so the conversations went on. And as it turned out, I ended up getting a job in San Francisco, and I moved there in 1980. So let me just shut this down here. Don't want to shut that, don't want to shut that. we'll do this. Can go, okay. And so this is what happened. Got together, formed a company, and at UCSF at that time, there were two companies that had already formed, Genentech and Chiron. And people, venture capital people, would come through the halls of our department looking at posters on the wall, and they'd grab you and say, geez, do you know any technology that we might turn into companies? So we met some people who had a diagnostic company, Parker Montgomery. And he said, we told him about liposomes. He said, why don't you come down and talk to us? And so we did. We went down there on Memorial Day. And we talked to them for 4 hours. And we walked out of the room on a handshake. They gave us $2.9 million. So no business plan, no elevator pitch, no going around to venture people. Good old days. Here's the money. Let's make products. So we made products in the diagnostic area within the first 6 months, and we decided that we would go ahead now and we could run a company. And so what I guess I would tell you is that I learned business practices the way I learned about sex. It was on the street. So no classroom, no accounting. I could hardly balance my checkbook. And so we started this company, Liposome Technology, in 1981. It was based upon the RAV patents. And in 1984, we thought we were really good in 1981. It took us another 2 years to raise money for the drug delivery company. And so then it became a little bit harder. In 87, we went out on an IPO, and you know what happened at the end of 1987? The market crashed, and so all of a sudden our company is struggling for money. And we started the doxorubicin project then. And that project really was based on research that had been funded by NIH in the late 70s, early 80s at Roswell Park. We got a product approved, Amplitaq, in 1992, and that product actually came out of my laboratory. It was funded by NIH in the early 80s from an NIH grant. And in 1982, February, we have 1 month's salary left. And we met David Bleck. David Bleck was, at that time, the early 90s, the guru of the biotech industry. He had founded ICOS. He had founded earlier a diagnostic company, the first one to use monoclonal antibodies that was sold to BMS. And David Bleck was Mr. Biotech funding. And so he came in, believed in what we were doing, put up money, and it saved the company. And we were able to go on and get Doxil to the FDA and get it approved in 1995. Now, if you look there, we got it approved on 70 patients. And I'll tell you a little bit more about that in a few minutes. In 95, it was approved for Kaposi sarcoma. In 99, it was approved for treating ovarian and multiple myeloma. In 99, the company was acquired by Alza. And Alza, in turn, was acquired by Johnson & Johnson. Okay, so that's sort of the history. And if I learn how to run this, this is the product. It's used to treat cancer, and it's a nanomedicine. And it's a nanomedicine, and that means it has a dimension smaller than 100 nanometers. And this length scale here shows the products, the red blood cells, drug delivery device. We have hemoglobin in the red blood cell. It circulates through your body, and the drug it delivers is oxygen. The liposome is about almost 2 orders of magnitude smaller. It's 100 nanometers. And this is a cryo-EM image of the liposome. You can see what I'll show as a bilayer, and that's the drug inside of it. And then down here is a typical low molecular weight drug. This happens to be a chelator, DTPA. So this is the length scale that we're dealing with. Everybody in the audience has made a liposome. So how many here scramble eggs? Everybody raise your hand. Is there anybody in the audience who's never scrambled an egg? I don't see anybody's hand. So that means all of you have made liposomes. When you scramble an egg, what you're doing is you're taking phospholipid, mixing it up with the aqueous phase, and if you put that scrambled egg before you fry it under an electron microscope, you will see a liposome. And a liposome is this lipid vesicle that has a lipid bilayer surrounding an aqueous phase. And it's, as you know, it's very small, but if you took a milligram of lipid, a very small amount, 1 milligram, and you turned it into a big liposome, it would be the size of a basketball. So if you can imagine this basketball here with a very thin membrane, and we can put drug inside of it. And the drug that we put inside of it is doxorubicin. So this is the drug, a widely used anti-cancer drug. We put the drug inside of it. We encapsulate it in the lipid bilayer, and on the outside, we put a polyethylene glycol coat. And so what we did as scientists and in the company is we invented 3 technologies. One technology is the method for producing the liposome at high volume. It was an extrusion technology. And that came out of a paper that was published in 1978. Another technology was the polyethylene glycol coating, and that allows the liposome to circulate for a very long time. And the third technology was a way to add the drug outside of the liposome and have it go across the bilayer and become concentrated and precipitate inside the liposome. So these were 3 inventions that were enabling for this product. And here's just one image of one of the Kaposi's sarcoma patients imaged with indium DTPA, which is inside the liposome. And this is a time scale, 4 hours, 24 hours, 48 hours, and 96 hours. And here, this is a lesion on the patient's leg. And you can see this lesion being imaged at 4 hours, and the contralateral leg shows no imaging, more intense at 24 hours, more intense still at 48 hours, and the drug and imaging agent is still there at 96 hours. And you can also see that the liposomes have gone to the bone marrow, they've gone to the liver, and to the spleen. And so the liposome has taken this drug, circulates through the body, and it's concentrated in the tumor. Okay, that's a long story. What's happened with this product? It sells about $600 million worth of drug a year. And in 2009, Johnson & Johnson had problems manufacturing it. And so there was a nationwide shortage with Doxil. It's made television, made the New York Times, and it's caused a lot of grief for Johnson & Johnson. Got Doxil. And the reason for this goes back to something that Bruce had sort of alluded to. There was a small company that could manufacture the product. The company is acquired by Johnson & Johnson. They got rid of everybody from Alza. They got rid of everybody from Sequis. There was nobody left in the company who was involved in the development of Doxil. So what's happened? The FDA has given interim approval for an Indian company, Sun Pharma, to import a Doxil generic. And a lot of other companies, there's 4 or 5 other companies that have Doxil generics. Okay, so a success story, new technology, it came out to a nice ending. In 1994, I went to Houston and I became involved with 7 other prominent scientists to start gene medicine. So here we are. This is the Astrodome in Houston. Our first company was right up here on North Stadium Drive. We had a charismatic CEO, Eric Tomlinson. he had been at Novartis, and he was the best CEO, startup CEO I've ever seen. Highly efficient, very persuasive, and there was a great cell biologist, Bert O'Malley Sr., and Robert Schwartz from Baylor, Savio Wu, Dennis Roop, you know this person here, Fred Ledley, and one of the first hires was Alan Roland, who was working at the company. And so I went down there for 9 months as the chief technical officer and ran the science until I hired my replacement so I could go back to the university. And our mantra was, the patient will make their own medicine, and that will dose genes the way that we dose drugs. The idea being that we take a gene, maybe a gene for factor IX, put it in a delivery system, and that will introduce it into a cell. The cell will take that information in the form of DNA into the nucleus, would make messenger RNA, manufacture the protein. And so in one sense, it's almost like holistic medicine. The dose of genes that we're giving is relatively small, and we expected the patient to make their own medicine. We were an early company, and we had fantastic science, but it turned out that two things happened that from the financial side were bad. There was a very, very highly publicized death of a patient at the University of Pennsylvania in an adenoviral trial. And this totally took the air out of the gene medicine effort for all of the companies. And something else happened. The original funder and supporter of gene medicine was David Black. David had saved LTI. He took gene medicine, brought us to market. And it turned out that David had manipulated the stock. One of the companies he manipulated was gene medicine. And so this was a very nice business lesson for me. When your stock has been manipulated, investors are not so happy about investing in this company. And so we ran into a money wall, and we were acquired by our competitors. And so this was a bridge too far. So where am I going now? So it's a few years later. It's been almost 10 years since I've been involved in a company, and I've gotten back into the water with a company that's called Zone 1 Pharma, and this is in San Francisco again. And this is in the garage at UCSF. So during my career at UCSF, a number of interesting things have happened. When I first went there and we started LTI, many of my colleagues disapproved of having an academic involved in companies. In 2003, UCSF had generated a lot of money in biotech companies, and now they try to encourage the formation of companies, and they have a garage or a laboratory space that you could start a company in on the campus. And so we're in this garage. And our focus is something that would make a good target for gene therapy. That is thalassemia. And thalassemia is a disorder of hemoglobin. There are mutations in either the alpha or beta chain, and it leads to a shortening of the lifespan of the red blood cell, and they're rapidly eliminated from circulation. And it's thought to be a protective mechanism against malaria infection. And so around the Mediterranean, across the Middle East and India, up through lower China, thalassemia is more prevalent, and also in Africa. In regions where there are malaria, there's a higher incidence of thalassemia. And one of the consequences of thalassemia is the red blood cells are eliminated more quickly, and they appear in the spleen, so you have a distended spleen, among other things. For other things that happen is that when you saturate your iron proteins, in this case it's transferrin, you get free iron, and this free iron accumulates in the liver, the spleen, and the heart, as well as in the brain and certain endocrine organs. And it causes, probably the biggest toxicity is a myocardial toxicity. And so there are a number of approved treatments for iron overload disease as it manifests itself. There's an iron chelator that's infused into a patient 8 to 12 hours, 5 to 7 days a week. And so you have to carry a pump, and this product is infused into the patient. The other product, Xjade, is approved in this country, and it's given between 20 to 40 mg per kg per day as an oral drink. And both of these products are products of Novartis. So I've already shown you that liposomes go to the liver, the spleen, and the bone marrow, and this is just to reiterate that. Here we have liver, spleen, bone marrow, and this is a radio-labeled liposome in a person. And if you take a liposome encapsulated chelator and you use it to remove, in this case, it's plutonium. Plutonium accumulates in the same organs as iron does. And so this is the free chelator. This is the liposome encapsulated chelator, so it stays in the body much longer. And when you look at the elimination of plutonium from these patients, this is time and days. This is if the animal isn't treated. This is if the animal is treated with the free drug at 30 micromoles per kilogram. And this is if you treat the animal with a liposome at 3 micromoles per kilogram. So 1/10 the amount of drug, 3 times better elimination of the plutonium. And so we have a liposome that it could be very effective at removing a metal from the body. And so the way we like to think of our approach is the classical way is a chelator takes out iron one by one, and what the liposome will do is take iron out many at a time. So if we see that the harming factor in the body in this case is iron, the classical way is you have a wasp trap, and you get these iron molecules one at a time, and this is what desferoxamine does. What we think we can do is we can take the whole hive out at once. We can get rid of all the iron, at least for 1 unit of blood in 1 dose. And what we think the liposome desferoxamine will be able to do is give you unit for unit dosing. So when a patient gets a unit of blood, they'll also get a unit equivalent of a liposome chelator. Okay, so this is commercially viable because recently there was a Bay Area company that has a new oral chelator that was acquired by Schreier in a buyout. And so the market is big. People are looking at better approaches to remove iron from the body, and we think a liposome might be that. Okay, so what have in this very short period of time, what have I told you? I've told you about a number of different companies that I've been involved with many dynamic people over the last 30 years. And the first one was LTI. And with LTI, we had a lot of invention that was enabling invention and a lot of innovation. And we came into the biotech sector at the right time. It was lucky. We were lucky. We were lucky to get our product through the FDA. Even though the science was great and the clinical trials were well done, it was always, we were lucky. The second company was gene medicine. We had even a bigger brain trust and a lot of technology. We started off gene medicine with more patents than we had started LTI off with. And we innovated. We did actually 6 clinical trials, one for VEGF and 5 others for different combinations of cytokines. IL-2 was the main one. We also did an IL-12 interferon alpha combination. And so that company was just before its time. The technology wasn't good enough. And finally, our most recent effort is a company that almost is pure imitation. We're taking technology that is now off patent. All of the technology that we had developed, we and others had developed during the liposome early days, all of that is off patent. So we're trying to take that, we're taking ideas that have been in the literature but never moved forward, and we're doing them better, and we have some of our own patent protection, but basically I see this as imitation. And we think that we still might be able to do something that will be beneficial for patients in the near term with this approach. Okay, so there's a lot of people that have been involved, as you might imagine, with this. My mentor, Dimitri Papahagopoulos, was a great mentor. Nikorvanatides was relentless in keeping LTI and then Sequus going. Sally Davenport was Nick's assistant, who also was an early founder of the company, and she kept Nick going. The employees of LTI and Sequus. And we had a great retention of employees at LTI who went on to Alza, and then when Johnson & Johnson closed them down, they went on to other biotechs in the area. Zone 1 Pharma, there's 4 of us. We do have a Noble, so one of the founders is a Noble, Charles Noble, and the other founder is Mark Hayes, a biophysicist, analytical chemist, and Peter Working was at Sequus and was involved in getting Doxil and Amplitaq approved. And at Gene Medicine, it was just a very distinguished group of scientists, and Mark Gessler, who was an excellent CFO who later became the CEO of Genetics, Gene Logic. They are the employees, too, have stayed around. And NIH, of course, for funding, and the University of California for the wonderful students and fellows that I've had in our group. And I think this is a good place to stop, and I'll be happy to answer questions.
Questions?
Thank you. A fascinating talk. I've got a question dealing around the ownership of intellectual property, especially early on in the collaborative process. A lot of people are involved. The research itself is really emergent. You're not really sure where it's going. There can be tremendous, outcomes through commercialization. How were those early questions handled? You said you were encouraged by your university to set up through your garage your own kind of, startup operation. How do you deal with, who owns what, how much goes to the university, how much, is shared by collaborators, many of whom come from different disciplines, even different institutions?
Okay, I'll answer that question from my perspective. We are fortunate to have 2 very knowledgeable lawyers among us, and they can correct me in all the things that we do wrong. So the first thing is that in our group, I patent everything through the University of California. And so it turned out that with LTI, Dimitri and I revealed our inventions to the Research Corporation, which patented it for Roswell Park. Roswell, this is 1978, they said, we don't think liposomes will ever be drugs. And so we said, fine, give us the patents back. And they did, and we found a very good lawyer in New York who filed it was one application that was divided, and it came out, so we had 5 applications that were approved that turned into patents. One of those patents was the first patent for gene therapy that was issued by the U.S. Patent Office, and that was issued in 1984. And one of the patents was for pesticides. And so we're out raising money, and we had a investment banker in New York. They see the pesticide patent, they hook us up with Union Carbide. And here's four people don't really do business. We have this investment banker who really wasn't a very good investment banker. He takes us to Union Carbide. And I can remember driving down the New Jersey Turnpike, and in those days, everybody smoked. And I'm with three Greeks, and Greeks really smoked. And we're driving down the New Jersey Turnpike. The car is smoke coming out. We get to Union Carbide. We're late. They're waiting for us. They take us to the conference room. We walk in. The table in the conference room was from here to the back of the room, and around it was all their lawyer and corporate people and vice presidents. And we hardly knew what to say, because the only thing they wanted to know was, have you done any field work with your liposome encapsulated fungicide? No, we hadn't done anything. You know, we were in Cancer Institute. But that's one way to handle intellectual property. University can give it back to you. What usually happens is the university evaluates it and decides if they're going to go ahead and file a provisional or an application. And what they do is they will interview the people in your lab who have worked with that invention, and we have the lawyer decide who the inventors are. So that's the way we do it at UCSF. And then those people that are considered to be inventors are put on as inventors. And if it turns out that you keep improving the invention, if other people add inventive aspects of it, they can be added later. That's the way it's usually done, and that's the way we do it. And so in my laboratory, what I do every year is I have the lawyer from the tech transfer office come to my group without me there to describe what the process is, so that everybody in the lab knows that we like to patent things, we like to move things forward, because without a patent, there's a reluctance many times to move a medicine forward. That's the way we do it. I don't know, Bruce, you may have more to say, or Alan.
Well, there may be another piece to your question. I think what the federal government did in 1984 was make it possible for universities, even if they receive federal funding, to own the inventions. And the next step would be then, if you did a transaction with a company, the university would sign an agreement with the company. And sometimes the members of the lab would become employees of the company, but it would be a university-company transaction. I don't know if that was the other half of your question.
Well, especially when you might be talking about multiple universities involved, as research really does become collaborative across different units, which can complicate it if each has a slightly different approach to it.
Yeah, what usually is done is there's an inter-university agreement, and they decide if these are the inventors, which university will take the lead on the patent. And so you get this agreement between the universities. And so we have one in place between us and Stanford. Yeah.
Next question, Mike.
Hi, I'm an undergraduate student here at Bentley, and I just had a question about the business aspect. You were talking about nanomedicines and liposomes and phospholipids, and here at Bentley, a lot of us really don't have a background in any of that stuff. I understood the concepts of what you're saying, but I really didn't understand what you were saying. So how does an accounting or a finance major at somebody at a business school such as Bentley find success in a biomedical company or in the field of science? Is it even possible? Is the barrier of knowledge kind of too strong to find success?
No, I think that in the companies that I've been associated with, the people that are coming in on the financial side often come in with strong financial background. And they're not scientists, and they might not have even had a great focus on science as an undergraduate. And in an open company, in a small company, you can't help but be exposed and pretty much be brought up to an understanding of what the company is doing. In the early days of LTI, we had 12 people. It grew to 45, and still everybody there knew what was going on. The accountants would come to the seminars. At gene medicine, we grew to maybe 125 at its biggest, And when it got to 125, then communications became a little more disruptive. But in a smaller company, scientists will include you in the discussions of what's going on. So I don't think, if you bring in strong business skills, I don't think you have to worry about being out of place in a biotech or chemical company. Yeah. Yeah.
I saw that you have had many successes in medicine, but have you ever created a product or a medicine that turned out to be defective? And if yes, any client filed a lawsuit or anything for the medicine?
Lawsuits are unavoidable in medicine. There's never been a class action lawsuit against Doxo or Amplitaq. But I'm sure that there are some lawsuits that may involve with how the drug is administered or other mishaps that would be at the local level. But there hasn't yet been a product recall of the two products that I've been involved with. And so we've been fortunate of not being involved in any of that.
Frank, if I can just add,
Yeah.
and I think the question addresses the fact that we deal with drug products here, and there is no drug on the market. Doesn't matter if it's aspirin or Tylenol, there's no drug that doesn't have side effects. And in the law, as you know, if you put a product in the stream of commerce, generally the producer is responsible for the costs of damage should it cause adverse effects, should it cause harm to individuals who use it. And I think what your question is getting at possibly is, how do we deal with that in the pharmaceutical industry? And there are two ways. One is that because public policy has found that it's so important to have drugs on the market, the rules of tort litigation in the United States recognize the fact that some products are unavoidably unsafe. And the pharmaceuticals fall under that category. So that you have to really go out of your way and show negligence, gross negligence sometime, for a producer. But the surprising answer, and I'll say more about this later, is that the Food and Drug Administration, the FDA, that so many people think stops progress of new products, actually provides cover for the industry. Because FDA, following laws passed by Congress, has developed regulations for the appropriate and safe development of drug products. And companies can refer to those regulations and use those as affirmative defenses as long as they follow FDA regulation. So, in a way, the government, through Food and Drug Administration, provides a cover for the problems that you talk about.
We have to move on, but we'll continue these great discussions in our next section. Thank you, Dr. Szoka.